This is a lecture continuation of cell structure and function, and today we're going to talk about movement through the plasma membrane. We're going to look at different ways that substances are transported from one side of the cell membrane to another. We can summarize the major pathways which substances take across the membrane by 4 major processes, and that is diffusion, osmosis, filtration, or mediated transport mechanisms. And we'll specifically talk about facilitated diffusion, active transport, and secondary active transport. So the diagram here shows the plasma membrane. If you recall from the previous videos that the plasma membrane has these hydrophilic or water-loving phospholipid heads which attract water-soluble substances, but the tails are hydrophobic, meaning water-repelling, and water-soluble substances cannot diffuse directly through this inner core because it's a fatty layer. So we know that water and oil don't mix, so picture this as an oily core that will not allow water-soluble substances to pass through. So the only things that can pass through would be those things that dissolve in oil. For example, the different gases, carbon dioxide, oxygen, alcohol, cholesterol, hormones that are lipid hormones. They're fatty substances such as estrogen, testosterone, your fat-soluble vitamins like vitamin D, vitamin E. Those things are things that will dissolve through the membrane that don't need to be specially transported across. Things that are water-soluble, on the other hand, do need to be transported. So here we see there's special protein carriers to allow those substances to cross that fatty core. And we'll talk about the special transport mechanisms that involve these protein carriers. So the first process we're going to talk about is diffusion. And the key thing to remember with diffusion is that it's a movement of substances. When we say solute, solute is any substance that is dissolved in a liquid. So any water-soluble substance we could call a solute, as well as something dissolved in a non-polar or non-water-soluble solution that would also be a solute. So anything that is dissolved in a substance we call a solute. So what happens in diffusion is the substance that is moving across the membrane will move, if it can, from an area of higher concentration to an area of lower concentration. So as long as we have a concentration difference across the membrane, there will be a natural flow of a substance without the input of energy if it's able to cross. For example, you've seen this in real life. If you have a can of sardines, for example, and you open up that can, the concentration of gas molecules close to the can is much higher than compared to further away in the room. But those particles will continue to diffuse throughout the room until they're equally dispersed and the entire room smells like sardines. So that's just a simple example of diffusion. So how easily a substance can travel from one side of the membrane to another depends on how much of a difference there is in concentration. We call that a gradient, a concentration gradient. So if we look at the previous slide, we can see there's a higher concentration of this substance on this side than there is on this side. So therefore, these substances will always diffuse without energy from this side of the membrane to this side of the membrane, as long as it can dissolve through that fatty core. So this might be a substance, for example, that's fat soluble, say, example, one of those hormones like we said, or alcohol, for example, can dissolve in a fatty core. So it would flow from high concentration to low concentration. So the more particles there are here and the fewer there are here, that would be a steeper concentration gradient, and we'd see more ready diffusion across that membrane. Another thing would determine how easily that liquid flows. So the more watery the substance is, the more easily it will flow. So high viscosity is a thicker fluid and low viscosity is a thinner fluid. For example, molasses would be a high viscosity substance versus, for example, a cheap pancake syrup that is a little watery. That would be a low viscosity substance. So the cheap pancake syrup would be able to dissolve more easily than, say, a thick molasses would across a membrane. So again, no energy required for the process of diffusion, no transporters involved. It's just the movement of substances from an area of higher concentration to lower concentration on one side of a membrane. Osmosis is very similar to diffusion, except we're talking about water. So it's specifically the diffusion of water. So don't confuse that with any other type of diffusion of a solute. We're talking about water moving. So when we see water move from a dilute to a more concentrated solution, that is osmosis. So there's 3 different terms that we use to describe a solution, telling us what the concentration of solutes is in that solution. So in this example, this is a hypotonic solution. So that means we have outside this red blood cell a very dilute solution. So it's very watery, for example. So as a result, through osmosis, water will flow from a high water concentration to a less water concentration. So inside this red blood cell there might be more solutes dissolved in here, more concentrated. So the water is going to flow from more dilute into the more concentrated area. So we could see this red blood cell would eventually burst if we could not stop that flow of osmosis. Here we have what's called an isotonic solution. Iso means same, so that means we had the same concentration of solutes outside this red blood cell as well as inside the red blood cell. So we'll see a little bit of movement of water back and forth, but there's not going to be any net movement in one direction, so this red blood cell stays the same. And this is what our body fluids are like, and it's the same thing if you're just trying to maintain the hydration levels of a patient. We want to make sure whatever IV fluids we hang are isotonic with the patient's body fluids. That way they won't lose any water or they won't become puffy and gain too much water in their body tissues. This is an example of a hypertonic solution. Hyper means we have lots of solute, very concentrated. Maybe this is salt or sugar. It's very concentrated outside this red blood cell. And then inside the red blood cell, it's less concentrated with salt or sugar or some other solute. As a result, water by osmosis is going to flow from the more concentrated side to the less concentrated side. I'm sorry, from the less concentrated side to the, sorry, I'm saying this backwards, geez. From the less concentrated to the more concentrated side. So again, this is flowing of water. So this cell would shrink as it loses water to the environment around it because this is a hypertonic solution. So water always flows from where it's more dilute to where it's more concentrated. Again, here it was more dilute on the outside, less dilute on the inside, so water flows into the cell. Here it's more dilute on the inside, less dilute on the outside, so water flows out of the cell. And we see this happen when you pour salt on a steak prior to putting it on the grill. When you pour that salt on the surface, that is going to create a concentrated solution on the surface of the steak, and water is going to flow out of the steak cells in and form a little puddle on the surface of that steak. And as a result, that steak will be less juicy because you poured salt on it and pulled all the moisture out of it. So when we season our raw meats, we want to make sure we put a layer of oil over the surface, like rub it in olive oil or vegetable oil to prevent that water from flowing out of those cells and making your steak or your meat tough and dry. Another form of movement across a membrane from the blood into the tissues would be filtration. And this we see happening in the kidneys. When we have blood flowing into the capillaries that feed the kidneys, this blood is under pressure. And there's small spaces between the cells of the capillaries in the kidney forming little holes, and that blood flows out, or plasma specifically, flows out of the capillaries into these special collecting vessels in the kidney. So anytime we have movement of a fluid under pressure, that is an example of filtration. And there's an animation we can watch here that demonstrates what filtration looks like. Quick show that here. So here we can see the components of plasma. These probably are like glucose, sodium ions. Any of those substances are able to flow out of the blood into these special collecting vessels of the kidney. So it's blood under pressure. It's being filtered out and flowing from one membrane into another. So that's filtration. Then we get into these mediated transport mechanisms, which means they require a carrier. Now there's a special type of diffusion called facilitated diffusion where we have substances still moving from a region of higher concentration to lesser concentration, but there's no energy involved. There is a protein carrier that's involved, but there's no energy to drive this carrier. It simply changes its shape and transports that substance to the other side. So we see some glucose transport using facilitated diffusion. Glucose is water soluble, so it cannot dissolve through this fatty core, so it needs the help of a protein carrier to get it across to the other side. But again, notice we're going from higher concentration to lesser concentration, and this could be in either direction. If we had higher concentration of a substance inside the cell that needed to be transported outside the cell, it could use that carrier to go the other direction as well. But it has to be from high to low if energy is not required. And in the case of facilitated diffusion, the only thing required is a protein. It does not require energy in the form of ATP. Now this example is called active transport, and this is a special mechanism where energy is required. Here we can see ATP is donating a phosphate to this protein carrier. It's phosphorylating this carrier and giving it the energy it needs to move substances against their concentration gradient. So this pump is called the sodium-potassium pump, and what it does, with every cycle it transports 3 sodium out of the cell and brings 2 potassium into the cell. And these are against their concentration gradients, which means that we have a high concentration of potassium inside the cell, a lower concentration of potassium outside the cell, but this pump will continue to bring potassium in, going from low concentration to high concentration. And again, that goes against the law of diffusion, so therefore energy is required to transport these substances. So the same thing occurs with sodium. We have a high concentration of sodium outside the cell, but this pump continues to pump sodium out into the higher concentration from the lower concentration, and that requires energy. And we'll find when we study the muscular system and the nervous system that this difference in concentration and the action of this pump is essential to nerve and muscle function. Here we'll watch a little animation of the sodium-potassium pump. So here we can see 3 sodium going from inside the cell. ATP is binding here to the pump, giving it the energy it needs to transport those sodium outside the cell. Notice there's already sodium out there, so we're going from lower concentration inside the cell to higher concentration, which is against the concentration gradient that naturally flows high to low with the law of diffusion. Here we saw 2 potassium bound from outside, and they're being brought into the cell. So again, for every cycle of this pump, we're dumping 3 sodium out, 2 potassium in. Another type of transport is secondary active transport, and that's where we're using the pump here, which requires energy. We're dumping 3 sodium out, 2 potassium in, and that is causing a buildup of sodium outside the cell. Then we have a special carrier molecule further down the plasma membrane where sodium is driving the pump to change its shape and bring sodium inside the cell. And this is a high to low, so that does not require energy. But glucose is going from low to high, which normally would require energy, but because it's using the energy from sodium and its concentration gradient, glucose does not have to expend the energy directly. So the energy that's being used here is driving the pump to get more sodium out, but glucose is just riding along with sodium on this protein carrier. So we call it secondary active transport, which means glucose isn't really using fuel. The sodium-potassium pump is to get a gradient going, and then sodium is flowing in. So it's kind of like a taxi driver. So the taxi driver pays would be like sodium, pays the energy, the ATP, to get outside here, and then glucose just hitches a ride along with sodium into the cell, going against its concentration gradient. And the last few processes that we're going to talk about all require ATP, and they're a little less complex in how they work. So when you talk about whole cells here, the plasma membrane can transport whole substances in a very nonspecific way, which is called endocytosis. So many of our white blood cells are called phagocytes, and phagocytes means cell eating, and that's where the plasma membrane will surround a substance. In this case, this is a bacterial cell. It can surround this bacterial cell and engulf it, form a vesicle around it, and then release enzymes to break down that bacterial cell. So this is an example of phagocytosis. Phagocytosis means bringing solid materials into the cell by wrapping the plasma membrane around it and forming this vesicle. Pinocytosis means cell drinking. So anytime the cell might be taking in a liquid material, we call that pinocytosis. Again, bringing it into the cell. And another type is called receptor-mediated endocytosis, which means on the plasma membrane are special receptors that the substance has to bind to first, and then it's brought into the cell and a vesicle is formed. But if it doesn't bind to the receptors, then it's not brought into the cell. And the opposite of endocytosis is exocytosis. So if we have, in this case, we have an organelle that is producing a substance, wrapping it with the membrane around the organelle, and then continuing toward the plasma membrane and fusing that membrane with the plasma membrane of the cell and dumping out that substance. So exo, think of exit, it's leaving the cell. So we see this process occurring in cells of the pancreas, producing digestive enzymes and these vesicles, and secretion of mucus by the salivary glands and milk by the mammary glands is all exocytosis from those cells, producing the substance and releasing it from the plasma membrane. And that concludes our discussion of cellular transport mechanisms.